Hello. If you're watching this video, you more than likely are one of my anatomy and physiology students. And today's video is going to be covering the RAAS, which is referred to as Renin-Angiotensin-Aldosterone system, which is a system that the body uses to raise blood pressure. It's a blood pressure control system, but the emphasis is on raising the blood pressure. If you are watching this video, you should already have a fairly good understanding of the renal anatomy, in other words, the anatomy of the kidneys, and pretty much how the filtration system operates. So keep that in mind if you're viewing this video. You should already have a good understanding. You should have studied the urinary system. You should have studied the lecture presentation notes that I've given you, and have a firm understanding of how the urinary system works, filtration system, glomerular filtration, flow, and the rate of filtration, and all those concepts, all those related concepts. Otherwise, some of the stuff I'm going to be talking about here may be a little confusing. So please bear that in mind if you are watching this video, that you should already have an understanding of how some of the physiology already works with respect to the renal system or the urinary system. If you're not one of my students, let me introduce myself to you. My name is Gary Senia, and I'm a professor of biology here at Suffolk County Community College in Long Island. And at some point in this video, at the end, I will post my email. Please feel free to email me with some questions you may have, or if you have any comments you'd like to share, I certainly would welcome any of your comments. Okay? All right, why don't we first get started with this general understanding or idea of what blood pressure is and why it's even very important. Blood pressure is extremely important for human physiology because it allows for tissue spaces, it allows for cells and tissues to have access to blood. Without proper pressure, without proper amount of pressure, blood flow is not going to be sufficient. It's not going to be able to reach various parts of tissue spaces, and cells will not have access to the blood. Without access to blood, the cells will be deprived of oxygen, deprived of nutrients. The cells won't be able to get rid of the waste products they have. So it's very, very important that blood pressure is maintained in order to perfuse the tissues properly. This idea of pressure, mathematically, blood pressure can be analyzed or looked at using a fairly simple equation, but I'm not going to really go into that here in this video. Perhaps in another video I'll talk about pressure and talk more specifically about the concepts or the relative properties of the components of this equation I'm talking about. But in brief, blood pressure is a relationship between the flow of blood and the resistance that the blood encounters during its movement through vessels. And by flow, I simply mean how much blood is being ejected out of the heart. Each time the heart contracts, each time the left ventricle contracts and ejects blood out of the heart through the aortic valve into the aorta, how much blood is actually leaving the heart, and how many times is the heart beating per minute. If we look at those two issues, if we look at those values, we can get an idea of blood flow, systemic blood flow. And then if we consider how much resistance that blood flow will encounter as it's moving through the vessels, we have this relationship. And this relationship is what we describe as blood pressure. Okay? But for our purposes here today, we just need to understand that pressure is an important issue with respect to the movement of blood and the ability of the body to perfuse tissues, and that it's directly influenced by several key factors. One is how fast is the heart beating? Okay, how much blood is being released out of the heart each time the heart beats, and with how much ease is the blood allowed to move through the vessels, so-called resistance. Resistance is something that the body can change simply by making the blood vessels, either smaller or larger. In other words, the vessels can constrict, and that's called vasoconstriction, or the vessels can get larger, dilate, and that's called vasodilation. And one of the mechanisms, or the primary mechanism by which the vessels dilate, get larger, or constrict, get smaller, is through the relaxation and the contraction of the muscle layer of these vessels, the so-called smooth muscle layer of the vessels. These contractions and relaxations of these muscle fibers are accomplished primarily through sympathetic response, sympathetic and parasympathetic responses. of the autonomic nervous system. So when the body requires, or when the body needs to raise blood pressure fairly rapidly, because the nervous system is a rapid response system. It's a communication system that delivers quick and rapid responses, or quick communication. When there's a need to adjust blood pressure in a rapid way, the nervous system can constrict, can contract, will cause the smooth muscle in the vessels to contract, and that's going to cause constriction, or it can cause the muscles, the smooth muscle layer to relax, and that's going to cause vasodilation. All right? So that's kind of like how the nervous system plays a role in the systemic regulation of blood pressure. What I'm specifically going to be talking to you here today about is this special collection of hormones, or collection of cells that through various different pathways share a common effect, and that common effect is to raise blood pressure. That's the effect of this RAAS. The RAAS is although there's several mechanisms that are used to accomplish this, the commonality there, or the common goal, is to raise blood pressure. So let's take a look at this RAAS. And as I said, RAAS stands for Renin-Angiotensin-Aldosterone system. And there are a couple of key cells that are players in this system. So let's take a look at it. The first up is JG cells. JG cells stands for juxtaglomerular. Let me write that out. Juxta glomerular. Juxtaglomerular cells. Juxta in anatomy means nearby, and glomerular refers to the glomerulus of the filtration unit of the kidney, meaning the nephron, and specifically the renal corpuscle. So these juxtaglomerular cells, they are located near the glomerulus. And as it turns out, looking at the filtration unit, we see that we have a vessel that comes into the the renal corpuscle, and a vessel that delivers blood out of the renal corpuscle. So blood comes in, becomes filtered, and leaves. The vessel delivering blood is called an afferent vessel. The vessel delivering blood away from the renal corpuscle is called an efferent vessel. And these juxtaglomerular cells, they are special smooth muscle cells that are mainly part of the afferent vessels. There are some JG cells that are part of the efferent vessel, but they are mainly part of the afferent vessel anatomy. And these JG cells, as I said, they're specialized smooth muscle cells. And when they are activated, since they're smooth muscle, when they're activated, they will contract. And when they contract, they will cause vasoconstriction, or the vessel size, the afferent arterial vessel size, will get smaller, and that will increase resistance and increase blood pressure. Okay? The second key cell is cells that are called macula densa cells. Macula densa cells are cells that are located nearby. They're actually located nearby to the JG cells, and they're located in the part of the tubule. The renal tubule includes the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule, right, the DCT. And these macula densa cells, they're located in the DCT, okay, in the DCT, in a region where the DCT is nestled in between the afferent and efferent arterioles. So if we look at the DCT in that region, right, this is the DCT, this is a cross-section of the DCT, and here's filtrate flowing. Those are macula densa cells. And what they do is they analyze or sample the filtrate as the filtrate is passing the DCT, as the filtrate is passing the macula densa cells. And what they're analyzing is, they're analyzing sodium concentration. They're analyzing sodium concentration. And if they detect a drop in sodium concentration, that's an activation stimulus. That stimulates these macular densa cells to secrete a substance that is classified as a prostaglandin. A prostaglandin is a group of hormones. These are locally acting hormones. Sometimes they're referred to as paracrine hormones because paracrine refers to a hormone that acts on nearby cells. And these prostaglandins that are released by macula densa when they are stimulated by low concentrations of sodium in the filtrate, what they do is they go and they act on nearby JG cells. That's why they're called paracrine hormones, because they're acting on nearby cells. The nearby cells are the JG cells, and they stimulate JG cells. Anytime a JG cell is stimulated, whatever the stimulus is, when JG cells are stimulated, these smooth muscle cells that are part of the afferent arterial anatomy, they will release a substance called renin. Okay? So, JG cells are key cells, macula densa are key cells. Some other key cells that are part of the system are liver cells. Liver cells, okay, they're going to be involved because the liver produces a protein, a very large protein called angiotensin-aldosterone. Angiotensin-aldosterone. It's a huge protein. It's about 500 amino acids or so. And it's produced by the liver on an ongoing basis. It's inactive, which means that basically it just kind of circulates like as if it's asleep. It doesn't really do anything. It's there, okay? And this angiotensinogen is waiting to be acted upon by some enzyme. And that enzyme that's going to actually act on it is an enzyme, renin, that the JG cells will produce when activated to do so. So let's keep that in mind. So we have JG cells, macula densa, liver cells that produce this substance that's going to be part of the overall group of reactions that make up this RAAS system. Adrenal cells refer to cells that make up or are part of the adrenal gland. The adrenal gland is located anatomically above each kidney. So we have two kidneys, we have two adrenal glands. And the adrenals actually have two regions of cells, a medullary region and a cortical region. And the cells that I'm referring to are the cells that are part of the cortex. These adrenal cells, they're going to manufacture a hormone called aldosterone. And aldosterone is, as the name is seen here on the slide, is the third player in this RAAS system. Okay, so these adrenal cells are a key cell because they're producing a key player. All right. So what will stimulate these adrenal cells to produce aldosterone? One of the other products that are going to be formed from angiotensinogen, which I'll get to in a moment. Okay. Another key cell are sympathetic nerve cells. Sympathetic nerve cells are part of the autonomic nervous system, and when activated, like during that system-wide regulatory control process that I talked about when I first began this video, when the body needs to regulate blood pressure, specifically when it needs to increase the blood pressure, one of the ways it can do it is it can stimulate a quick, rapid motor command through the nervous system, which will stimulate the JG cells. And again, anytime the JG cells are stimulated, they will produce a product or a substance called renin. All right? So let's start with that again. JG cells, they produce an enzyme called renin. Renin is an enzyme which means it's a biological catalyst. It's going to catalyze reactions. Okay? So whatever stimulates a JG cell, whether it be stimulation from macula-densa prostaglandins, whether it's stimulation from sympathetic nerve response, okay? Whatever the stimulus, renin is going to be produced. Once renin is produced, all right, it's going to circulate, and whenever it meets up with an angiotensinogen, which is constantly being produced by the liver, is in constant supply within the circulation, whenever renin meets up with angiotensinogen, it's going to form something called angiotensin-1. Angiotensin-1, which is a precursor for another substance, all right, which is going to actually be a substance that's going to cause increase in blood pressure. So angiotensinogen is converted to angiotensin-1 by renin, and then angiotensin-1 is converted to angiotensin-2 by an enzyme that is also readily available, and that enzyme is called angiotensin converting enzyme, convenient, ACE. It's abbreviated ACE for convenience, or rather a convenient acronym is ACE, angiotensin converting enzyme. And that is located, ACE is produced by special endothelial cells that line blood vessels. And it used to be thought that these were only located in the lungs. In fact, some of the references you may come across that ACE is released from cells in the lungs, and that's a lot of where it is released. But lately it's understood that there are endothelial cells elsewhere in the body that also produce angiotensin converting enzyme. So to be complete about it, it's really not accurate to say that it's only produced in the lungs. So when ACE encounters angiotensin-1, it's going to convert it to angiotensin-2, and now angiotensin-2 is a bioactive hormone. The bioactive hormone, one of the properties of this hormone is it's a vasoconstrictor. Okay? So it can vasoconstrict. It can cause blood vessels, vasoconstrict. It can cause blood vessels to get smaller in diameter. In other words, the lumen can get smaller. If you increase or decrease the lumen, decrease the opening of the blood vessel, you increase the resistance and you increase the blood pressure in those vessels or in that vessel. Okay? So the property of vasoconstriction, okay, is an important property of angiotensin-2. But that's not the only thing it does. While it's vasoconstricting, it's also going to be traveling through the blood, because that's how hormones travel, right? Hormones are chemical messengers that travel through the blood. The nervous system is a communication system, just like the endocrine system, but the nervous system uses electrical impulses that are propagated through specialized cells called neurons. The endocrine system uses chemical messengers that that travel through the blood. So angiotensin-2 is a hormone, and this hormone travels through the blood, okay, goes to the adrenal gland. The adrenal gland has special cells in the adrenal gland that respond to angiotensin-2. In other words, angiotensin-2's target cells in the adrenal glands cause those cells to secrete aldosterone. Aldosterone, that's the third substance in this RAAS system. Aldosterone is a hormone. It's going to travel in the blood, just like other hormones, and its target cells are in the kidneys. In fact, it's in a portion of the distal convoluted tubule and the collecting duct, where it influences sodium reabsorption. So aldosterone will travel via the blood to the kidneys, and it will meet up with its target cells, which are actually in the last part of the collecting duct, so-called the late DCT. Distal convoluted tubule, the late DCT, the last part of the distal convoluted tubule, and the collecting duct. And what it influences is sodium reabsorption. Sodium reabsorption. In other words, it ramps up. It makes the kidneys increase sodium reabsorption, recaptures a higher amount of sodium than it otherwise would normally do. Okay? And what always follows sodium? Wherever sodium goes, water follows. So when the reabsorption of sodium is ramped up, water will follow. Water will increase the volume of the blood in the system, and that's going to translate to an increase in pressure. Okay? So there's a couple of things going on here. Lots of interconnecting mechanisms, but they all kind of share the same common goal, and that common goal is to raise blood pressure. So let me just kind of summarize this one more time. This RAAS system, which stands for Renin-Angiotensin-Aldosterone system, has several key cells that are involved here. The JG cell, juxtaglomerular cells, is a main player because it secretes renin, which kind of starts the whole process. All right? Renin is an enzyme, and renin is responsible for getting this whole activation system, or activating this whole system. So it's an important starting place. Renin is released from these JG cells, and they will convert whatever circulating angiotensinogen is around, which it's always around because the liver is always actively producing it. Okay? So whatever is around, it will convert to angiotensin 1. Once angiotensin 1 is formed and it meets up with its enzyme that will catalyze it to angiotensin 2, which is called angiotensin converting enzyme, ACE for short. Once angiotensin 2 is formed, angiotensin 2 has a property of vasoconstriction, so that will start to act on the system. That will start to vasoconstrict vessels and start to increase blood pressure. In addition, angiotensin 2 acts as a trigger for special cells, special cortical cells in the adrenal gland, which will cause those cells to release aldosterone, another hormone. Aldosterone will travel through the kidneys and act on its target cells, which are located in the late DCT and the collecting duct, and they will cause those cells to start ramping up collection, or reabsorption rather, of sodium. Along with sodium, water will follow. That will increase blood volume. That will translate into increase in blood pressure. In addition, we have the macula densa cells that are monitoring sodium concentration in the filtrate at the point of the DCT that happens to be nestled in between the afferent and efferent arterioles. And when the macula densa samples this filtrate and determines that the sodium concentration is low, by the way, let me make a point of that. One of the reasons why the body perceives that low sodium concentration means or translates into low blood volume and hence low blood pressure is because of sodium is, association with water. Every time you see sodium moving, water will follow sodium. So in the body's perspective, so to speak, if the sodium concentration in the filtrate at the DCT is low, that means the sodium concentration in the rest of the tubule is low, which means that the sodium concentration of the blood is low. In other words, because when the blood was filtered, right, and sodium was filtered out of the blood, if the concentration of the filtrate is indicating that there's a low level of sodium, that means there was a low level of sodium in the blood. If there was a low level of sodium in the blood, that means there was a low water content in the blood. In other words, the body views that as low volume, low blood volume, which means low blood pressure. So that's where the sodium blood volume pressure is connected. Okay, so getting back to macula densa. Macula densa samples the filtrate. If it's determined that sodium concentration is low, it triggers the release of prostaglandins. Prostaglandins are local hormones that act on the neighboring JG cells, which stimulate JG cells to release renin. And renin starts the whole ball, converting angiotensinogen into angiotensin 1. Okay? Again, the whole idea here is to maintain or increase blood pressure. All right? Again, thank you for watching this video. Hopefully you found it informative and helpful. Feel free again to send me any emails with any questions that you may have through my email, or if you have any comments at all, please feel free to share them with me. Thank you for watching.